Welcome back. So, I'm assuming that you've either watched my video on cell biology or you already have a grasp of cell biology. Now, in this course, we kind of want to look at the broader scale of cells, body cells. And body cells come from stem cells. So, today we'll be going over stem cells. Basically, their broader function in the body. So, let's make a little mind map so it's easier to follow along. Stem cells. Easy, right? What exactly are stem cells? Stem cells are unique biological cells that have a remarkable ability to develop into many different cell types. Now, this is normally during early life, or, throughout the body, growth, or even repairing your wounds. Now, one thing that we really want to talk about when it comes to stem cells is potency. It's really important to go with potency because it kind of decides how strong the differentiation power in a stem cell is. So let's make a little branch of our mind map. Potency. Now, potency is kind of referring to the cell's ability to differentiate into different cell types. Now, there's different types of potencies when it comes to stem cells. And, during GCSE, you've had like a pluripotent, multipotent, but there's kind of some more advanced definitions that we want to look at. So totipotent. A totipotent stem cell can differentiate into any cell, including the placenta. So let's write it down because that's important. A totipotent stem cell, it can differentiate into any cell plus the placenta. These are normally found during the early life of a human being. So, basically around when the zygote starts forming into, the embryo. So they are like taken from very early like embryos, basically. Now we want to talk about another form of potency in stem cells, which is pluripotent. You probably had a pluripotent, but there's going to be an extra part of the definition that you need to really pay attention to when it comes to a pluripotent stem cell. So let's make another branch. Pluripotent stem cells. And they can turn into any cell excluding the placenta. So differentiate into any cell excluding the placenta. That is a very important difference between totipotent, I forgot T, a totipotent stem cell and a pluripotent stem cell. Now, a multipotent stem cell, another form of potency in stem cells is a multipotent stem cell. So let's do multipotent stem cells. Now, multipotent stem cell can differentiate into a limited range of related cells. Now, what I mean is, stem cells found in bone marrow can turn into different kinds of blood cells, basically. So they can differentiate into a range of related cells. And lastly, we have a unipotent stem cell. They can only really, differentiate into a certain type of cell. Like, a good thing about unipotent stem cells is that they can renew themselves, which is really important when it comes to wound healing, especially when it comes to, scrapes on your skin, for example. For example, you get a little scrape on your knee. A unipotent stem cell, like maybe a unipotent skin cell, can kind of patch it up with a big bandage. So unipotent, one type of cell, but can renew itself. Oh, what? Oh, but can renew itself. Now, that's kind of going over all the potencies that you'll see in stem cells. Now, when I seem to be mentioning the word differentiation a lot. So let's go over the definition of differentiation. So let's make a different branch of our kind of mind map. Differentiation. Now, what does differentiation exactly mean? Differentiation is the process where an unspecialized cell becomes specialized. This is achieved through selective activation of genes within the cell. So let's write that down because this is a very important definition. So the process where an unspecialized cell, meaning a stem cell, becomes specialized. Usually done through the activation of a gene. Through gene activation. Now that we know the definition of differentiation, we can move on to the types of stem cells. We went over the potencies, but we haven't really went over the types of stem cells that you can encounter when we are discussing stem cells in this course. So, types. I explained it because it's very important. Okay, so first we have embryonic stem cells. These are found in early stage embryos, the blastocytes. They are pluripotent and divide indefinitely. So, I'm just going to sort it down to ECS, embryonic stem cells, found in early embryos. Now, the reason why these are kind of sought after is because they can divide indefinitely. So it's really good for research, which we'll go over later. But keep in mind that embryonic stem cells are found in early embryos, and they are pluripotent, meaning that they can differentiate into any kind of cells apart from the placenta, and divide indefinitely. Divide indefinitely. Now, the second type of stem cells that I want to go over are adult stem cells. Oh, why did I write like that? Oops. Let's get rid of that. Adult stem cells. Adult stem cells are found in tissues like bone marrow, skin, and in the brain, and they are typically multipotent. They act as like a repair system in the body. So, adult stem cells are multipotent, meaning they can only differentiate into a range of cells, so limited, and are found in bone marrow, skin, and brain. And what do? They help with repairs. The reason they help with repairs is because multipotent stem cells they can turn into a range of cells. So the fact that they can turn into a range of cells helps it with these repairs. Because, for example, the scraped knee over there. For example, if you scrape your knee, the gene in the stem cells activates, and like skin cells are produced to help and slowly repair the wound on your knee. Now, the third type of, let me do a wonky branch. The third type of stem cell is actually quite a recent development in stem cell research, and that's induced pluripotent stem cells. Induced pluripotent stem cells. Now, induced pluripotent stem cells, it's basically where scientists program ordinary adult cells by activating or inserting a gene to return it, to kind of reverse it back into pluripotency. This is kind of a more ethical manner of creating pluripotent stem cells for research, and it's a recent technology, only came out in recent years, and I think, in my opinion, it's pretty cool. So, where a gene is introduced to return an adult stem cell back to pluripotency. There you go. That's a pretty big mind map. Let's go into detail about differentiating a bit more, because we've talked about the types of stem cells, we talked about potencies, we talked about differentiation briefly, but I kind of want to go into more depth. Differentiation is basically controlled by signaling pathways and transcription factors. Even though every cell in your body contains the same DNA, a muscle cell only expresses the genes needed to be a muscle cell. So, only the genes needed to be expressed, unneeded genes remain dormant or, silenced. You can basically say that it's a matter of gene regulation. Differentiation is effectively a process of switching off genes that aren't needed for the cell-specific function. You really wouldn't need a skin cell that has the properties of, cardiac tissue. You wouldn't need that. So now that we've gone over the differentiation in a bit more depth, the potency and types, let's go over how it can be used in a medical setting. So let's go over here, because we don't really any space there. Medical application and research. Medical application and research. Stem cells offer massive potential for regenerative medicine. So think about Parkinson's disease, where, the neurons in the brain are degrading. With the technology that stem cells provide, we can research different ways to kind of regenerate those neurons in the brain and kind of help, patients live longer and fulfilling lives. So, massive benefit to regenerative medicine. It can also treat diseases. Like I said, it can replace and regenerate neurons, and replacing damaged cells in conditions, whether like diabetes type 1, where the body doesn't produce insulin or doesn't produce enough insulin, we can use stem cells to create insulin. Or repairing spinal cord injuries in people that are paralyzed. So it's a massive boon to regenerative medicine. It can also help disease modeling. So it can help disease modeling by growing tissues in labs to study how diseases progress. So, for example, how we can also kind of model things like antibiotic resistance and how to create more variations of medicines to help combat that, basically, as well. So, growing tissues in labs to study how diseases progress. Another thing is drug testing. Drug testing and basically testing the safety and and efficiency of new medicines on human tissue in dishes rather than animals, which is a great like ethical consideration when it comes to stem cell research and when it comes to, medical research, when it comes to making drugs. So, drug testing. So, testing the safety and efficacy of medicine on human tissue. Now, with all medical research, and while the idea of stem cells seems really great, there are some really great ethical concerns and considerations that you have to make before going into things like stem cell research. Specifically with the use of embryonic stem cells. So let's make a new section for ethical considerations. Because with any medical research, you really do have to consider the ethical repercussions. Ethical considerations. So the use of embryonic stem cells is actually quite controversial because, embryos can't exactly consent. And to use embryonic stem cells, you have to destroy embryos. This is a bit problematic, but if we look at it from a scientific standpoint, the embryo isn't really living a life. But if we look at it from a moral standpoint, it's pretty bad. It is pretty bad. So the use of embryonic stem cells is inherently problematic. But the problem is, if we take from adult stem cells, we can't get the proper benefits of using stem cells because adult stem cells are inherently multipotent. They can only turn into a range of cells, when embryonic stem cells can have totipotent properties or pluripotent properties, which we would need to kind of cure certain diseases or kind of investigate therapies for certain diseases and disorders. So it's problematic. Embryos can't consent. And honestly, and I think I've pretty listed most of the concerns. And also, there is the risk of playing God. When we are thinking about altering something, even when we're talking about maybe gene therapies, we are inherently going to play God. Someone with Parkinson's, for example, that's how they were kind of made. That's kind of how it kind of happens, bro. But what we also kind of have to consider is, does the person want us to fix the problem? And, it might sound silly because, of course, you'd want to fix that problem. It's affecting their life. But if we do this one thing, what stops us from making, genetically manufacturing babies or giving people more desirable traits using stem cells? It's a very slippery slope, and it's still actually being discussed in the medical world. However, there is induced pluripotency. So, and this is where, I already talked about this. They kind of turn it, they make it pluripotent they make adult stem cells pluripotent. And this is a lot more ethical because the adults can consent. And it's a lot better for trying to investigate curative therapies for different disorders. But again, it still poses the risk of playing God. But so does anything with medicine. Anyway, thank you for watching my video on stem cells. I hope this helped you revise, and I'll see you later.